Okay, so thank you to attend this this WheatIS half-yearly meeting. I will share my screen. And full screen. So this session will be recorded, but it will be only for the expert working group internal use. So feel free to present some preliminary work, and feel free to have a discussion on ideas, to share ideas, and so on and so on. It will not be published on YouTube or so on and so on. It's just for us, and also to the other expert working group members who will not be able to attend. So the quick agenda for today's meeting. So a quick welcome from me, and then we'll have a presentation of Raphael working with me at INRAE, and he will present you the test we want to do with BioCipher and BioChatter on our knowledge graphs. After that, we'll have the presentation of Sarah about AI from EMBL, EBBI. And to finish, there will be the Q1's presentation and literature review in NetMiner. Unfortunately, Tanner just sent an email. He's sick, so he asked me to postpone his presentation about AI in grain genes for the WheatIS annual meeting at PAG. And also, Dave, I asked Dave to postpone his presentation because of time zone issue, because in Australia it is very difficult to connect the afternoon, and it is better for us to do all our presentation in one slot. So he will also present his LLM update from University of Western Australia in PAG. So the main goal is to share ideas about AI, and so feel free to ask questions. I encourage as much discussion as possible. And I think we can interrupt the presenter to ask questions also if needed. And just a quick announcement for the WheatIS annual meeting at PAG. We already started to discuss about that. So it will be Friday morning. So we are in the process of booking the room and paying the room and the audiovisual material. So please travel on Thursday so you can be on site on Friday morning. And that's okay for this quick introduction. Do you have a question about this intro? If not, hello, Laurel. If not, I will give the microphone to Raphael.
Okay. Can you hear me?
Yes.
Okay. And do you see the slides?
Yes, perfectly.
Okay. So hello, everybody. So I will speak about some perspective project we have next month, exploring BioCipher and BioChatter frameworks for enhancing knowledge graph interaction. Before starting with this framework, I just make a small state-of-the-art of our internal graph knowledge graph development. So this is a work that has been published last year. It's a work by Baptiste Imbert and colleagues. It's a Neo4j database which has been implemented to integrate data related to legumes with ontology between 5 species on grain legumes. So what is displayed here is the metagraph of the Neo4j database. So Neo4j, I suspect everybody knows what it is. It's a graph database If you have any question, do not hesitate to ask. As you can see here, there is several color for group of nodes. So this is a AutoKB framework with it's a translational database model consisting of 29 core node types. Each one is represented by colored circle, indicating different entities with labels and properties. And key categories include in blue here, gene, RNA, and protein nodes. The move on this part concern homology and centenary nodes. So he has developed a framework, a pipeline to integrate data and making analysis among different genomic information from each species. In yellow, he has integrated several ontologies, such as the plant ontology, the PECO, the Go, and the MapMan ontology annotation term also. And in yellow, in dark red, we have RNA data. And we have also a functional annotation in light green and the taxonomic nodes in light gray. And QTL data also, which has been extracted from different publication or from his labs analysis in orange. So this work has been integrated in the Neo4j database and it has been deployed on our platform using a Neo4j browser. Some of you know it also. I put the link of the tool in the slide. And it looks like that. So you have a simple browser with a query section you can use to query the database, and WheatIS has developed a guideline to explore the data with a specific use case. So we have some guides which is visible on this part of the screen, and a user can switch on the next part to explore the data with an opinionated view with a specific use case here. I won't describe it furthermore, but each square area contains a query which allows to search into the database, and the result is visible here in the form of a graph or on a table A4 according to the query of the selection of the user on this part. So this tool is quite indicated for exploring data with a very opinionated view and always knowledge of the query language, which is a cipher for the Neo4j database. It's an equivalent of SQL for a relational database. But this kind of tool needs to have a good knowledge of the graph. So there are other approaches we have explored, such as usage of Bloom. Bloom is a software developed by Neo4j, the company. It allows to prepare some queries and to explore the data in a user-friendly fashion, and allow to search for some kind of data. Here we can search for genes, for different RNA, and navigate into the graph with this tool, which is available as a desktop standalone tool for developing or for exploring data on a laptop or desktop. But if we want to use it for collaborative or to provide a service using leveraging this tool, we need to pay a license and fees according to Neo4j model, economic model. So it can give access to data, but it's not very useful for academic solution. So these tools are limited in different manner. It limits user expressivity due to customizing option or language, specific language knowledge in a cipher, for instance. Other approaches could consist of developing a specific visualization tool or query tool, which has been done for some of labs or group here, but it's very time consuming in development processes, and in general it's very specialist for particular needs. So there is a lack of adaptivity in the frame of needs. And if we look to models such as Bloom, Neo4j Bloom, it's rely on proprietary model needing licensing fees, and which restrict its usage for open science approaches. So there is a need for modular solution to accommodate diverse requirements. So in order to explore that, we plan to use a framework which has been published recently, which named BioCipher. This is a framework which uses threefold modularity that integrates resources and ontologies to create configurable knowledge graph. Secondary resources are transformed into task-specific graphs, which will eventually transition to primary resource adapter. And this common framework harmonizes data sources and enables complex queries across biomedical domain. And for example, it can connect mass spectrometry data to clinical annotation and genetic dependencies, which could be used for drug target identification or drug discovery, for instance. So this is a use case from the BioCipher framework, and we plan to test it in order to being able to use another part of the framework, which is named BioChatter. And BioChatter is a platform for biomedical application for large language model. The aim of this tool is to ease the interaction with integrated data and use natural language processing to query the integrated data. So the LLM assists with tasks like interpreting results. BioChatter works through primary response circuits, which craft prompts for the LLM and generate user response based on the conversation history, such as ChatGPT and other. And concurrently, there is a secondary circuit in this part, which performs tasks like database querying and fact-checking in a INRAE-URGI approach, retrieval augmented generation. And this platform is quite modular. It allows integration of specialist model for various tasks, which are orchestrated from the primary LLM. So it allows to leverage on several LLMs, such as ChatGPT from OpenAI, but also with other which could be installed on-prem, such as Olama. But apparently, it works better at the time with ChatGPT, OpenAI models. So to test those frameworks, we have organized a workshop in Versailles in next November, organized by Cyril Pommier, Sebastian Beyer, and Pedro Zubarros, and who has invited a BioCipher expert, Sebastian Lebetenzer from Germany. The objective is to build common workflow using BioCipher. And the breeding API, which is another standard in the phenotyping and the genotyping community in Elixir plants. And we have invited people from CoreCookDB, from NAO in Japan to work on those framework. Another objective is to deploy BioChatter on these knowledge graphs, such as a proof of concept for a user. and to improve capacity building for all attendees on knowledge graph, AI, and BioCharter, BioCipher specifically. The idea is to lower barriers for interacting with biological and biomedical data and agricultural data sets using knowledge graph and LLM. Integrate with BioCipher and breeding API. Use natural language queries to interact with integrated data and avoid the development of a complex visualization tool or and so on. And the other expectation is to benchmark and evaluate implementation of common queries between participants and adapt the queries by the framework for the agroscience domain. So that's a very exploratory events which is expected next month, and we will discuss, if you want, about use case you could expect for such framework, or if you have any other solution, let us know for building another workshop, for instance, later in next year, for instance. Thank you.
Thank you, Raphael. So feel free to ask question. Yeah, Q1.
Well, when in November is the workshop?
The week of the 11th, from the 11th to the 15th November.
Yeah, for instance, it's not open. It's only a test workshop with a small group of people from Elixir and NARO. But yeah, we plan to perhaps to make something more open. For example, if you or someone from your team are interesting, there will be follow-ups. That's the idea.
Yeah, but BioCipher is something I think, Marco, you mentioned in the past, say would be interesting to hear more about it.
I had a question in mind. I don't know if to make it here or maybe later in the discussion. So the major problem I had with BioCipher is on scalability. I even asked this once on their GitHub repository on the issue section. The thing is, we are used to build things like knowledge graphs and Neo4j databases in a progressive way. So by taking sources, loading them, mapping them in parallel, and in many cases also using things like a SnakeMake together with some cluster system, so that we can exploit multiple nodes automatically, because we have requirements that make that necessary. Like we have to load medium data points and create databases with tens of millions of nodes and edges. And I couldn't find at the time, like 6 or 10 months ago, that BioCipher can easily support that. I wonder if you have had the same experience or things have improved or something.
Okay, thank you for your feedback. We will test, and yeah, we can go back to you to if it's better now for the scalability. But yeah, to have someone with some experience on that, it's interesting for us, because yes, we will test, so we'll have the same issue as you probably.
It depends about the data we are working on, because I'm not sure we have a knowledge graph such big as Marco manipulates. So perhaps we won't reach thresholds, but perhaps there is improvement on the BioCipher side. We will be able to discuss with Sebastian when he will come to Versailles. I take notes.
Yeah.
Thank you.
Thank you.
And did you test also BioChatter, Marco, or you
No,
stopped your
that's
test?
one.
in BioChat?
No, we have other people working on things like LLMs. I'm not directly putting my hands on that. Kevin later probably will mention what we are doing with these systems. But yeah, personally, I haven't tried pipelines like building knowledge graphs and passing them to some LLM system like the one you presented.
Is your hope to build a chatbot interface for your Neo4j knowledge graph? Say, have you tried BioChatter yourself?
No, you're asking to me?
Yeah,
no. Yeah. No, it very. We didn't start any experimentation with this framework. We will start just before the workshop to prepare some data to be able to hack directly the first day. But at this time, I didn't try anything. So I can't say if it's very simple to manage, if it needs specific resources. If you want to install on-prem, for instance, what kind of resources it can require for being performant, for instance. So I don't know at this step.
I guess I was just going to add that the fact that it's been trained and kind of coming from the biomedical field, typically they have a lot more data than most plants can lay their hands on. So I would have taken that to be quite encouraging, that it should be scalable, but it will be definitely interesting to see how it performs and whether there's any limitations associated.
Yes. Good remark. Yeah. And Raphael, do you know if there's something, if there are chatbot in production using BioCipher and BioChatter in the biomedical era?
Yes, they have an example, an instance of BioChatter, but I didn't look at what it has under the hood, what kind of data it is.
Yes, it is a sandbox, or it is with real data and a lot of real data. Yeah.
They have some. They have some data, but I don't know what is under the hood. And they have two chatbots available. One simpler than the other, and an advanced one which can be used in the browser directly, and that is using a more recent JavaScript framework for manipulating, developing the web interface, for instance. And if you want to have some tests, you can look at next.biochatter.com. But I don't try it very much at the moment.
Yeah, it's very preliminary work. We'll test it during the workshop with real data. Is there another question or remark? It's okay for you. Okay, so Sarah, I will give you the microphone. Can you share your screen?
You just need to stop sharing. Thank you.
Yeah.
Organizing my bits of Windows. Alrighty, can you see my slides okay?
Yes, perfect.
Lovely. Okay, so I'm giving quite a sort of broad level overview of AI activities going on at EMBL and EMBL EBI. There are quite a lot of things to choose from, so hopefully there'll be some areas of interest to the different people in the call. So at EMBL, where our headquarters is in Germany, but we're kind of distributed across 6 sites, with the EBI being in Cambridge in the UK. We are a research organisation, so we're doing kind of research around discovery science and developing more kind of tools and production-ready things on the kind of research side. Where I'm based is more on the resources and services side, so we're thinking more about how we can make sure that our data is AI-ready, and we can curate that and make that available for community users. I'm also quite close to the training aspect as well, and thinking about sort of training in these areas and how it can both contribute to the AI field and also benefit from. So I guess the sort of main strength areas and things that we're focused on are really around omics technologies, images, and structural biology, and kind of ranging from all levels, from the molecular up to whole organism. So the imaging is quite a strong activity across EMBL. We have kind of, from light microscopy through to synchrotrons and quite a range of imaging going on. And there's lots of interest in how that can be kind of integrated into the development of experiments as well, which is quite kind of nice, how the AI-driven kind of analysis of images can help drive your experiment. We are kind of known for a lot of our omics services and data provision. And of course, we cannot talk about structural biology without mentioning AlphaFold, so I will have a few slides on that. So as I mentioned, a lot of what we do is about providing data at EBI, and so providing reference data sets that people can then use to train their algorithms and develop their systems. So really, provision of that data in a way that we can that is standardized and readily accessible, and kind of machine readable, and how we can add value to those resources through the experts that we have for curation and annotation. But where appropriate, we can use AI tools to help with that curation and annotation, as long as that's kind of flagged so that people realize that that's not coming from a human. But we can use those tools to speed things up and then add human review afterwards. So one of our kind of newest, if not the newest repository that we have at EMBL EBI is the Bioimage Archive. I like to mention it when I'm out and about because it is not so widely known as some of our other resources. But for across all various modalities of imaging, it's a repository where you can deposit that image data. Image data is quite large, so this is challenging in its own right to be able to host and accommodate that. But one of the areas that the team, the Bioimage Archive team, are working on is kind of how those imaging data can be provided to be ready for model training and benchmarking and then evaluation, all those kind of steps that are required. So lots of that going on in different parts of the imaging field. And one of the challenges where the imaging field is very diverse, it's rapidly evolving. There are huge numbers even of data formats, and just working with the various different formats, having standards for metadata. Well, there's kind of other activities that need to go on that therefore can make the data much more shareable and reusable and ready for people to train on. So they're really doing a lot of work in those communities with kind of computer vision and other groups to be able to provide that data in a way that's readily consumable and usable for this testing and training. So AlphaFold, I guess anyone who's been anywhere close to biology has heard about AlphaFold of late, particularly with the Nobel Prize. So this was kind of a collaboration. So Google DeepMind sort of developed the AlphaFold approach, and they were in collaboration with EMBL EBI, where we have the AlphaFold database that hosts all of the models that have been predicted. It was initially trained on a lot of data that came from EBI, so the fact that we have the kind of protein databases and all of the sequences and these protein structures and sequence data that have been kind of accumulating over many years and stored in standardized formats. So what kind of drove the development, or what was kind of input data that kind of pushed on to make these 3D structure models for like kind of huge numbers of proteins compared to what we had before. So we now have more than 200 million protein structure predictions. I think the number of experimental experimentally determined structures that we have in kind of PDB is in the tens to 100,000s rather than, you know, so the scale is like totally different. It's super popular, so we have many users and lots of API requests and an awful lot of citations in a very short period of time. And it's been used for a range of applications and drug discovery and diseases and much more broadly. So there's a lot of interest in these models and how they can be used in different ways. So what we do in Ensemble Plants, my talk is not very wheaty, but I have got a wheat example here. So we have a wheat transcript. If you're in Ensemble Plants and you come to or in anywhere in Ensemble actually, but you come to a transcript, you can access the protein model kind of predictions where we've imported them from the AlphaFold database and via our connection with Uniprot. So I wasn't brave enough for a live demo in the short time frame, but this is a kind of 3D, you can move it around on the screen. The nice thing about AlphaFold is it gives you confidences on the different regions. So the dark blue are very high confidence parts of the protein model, and the orangey are kind of much more disordered, much less certain what they really look like. And so we have this here kind of by default showing this sort of like confidence view, but we also have a bunch of other information that you can choose to switch on. And so down here, if we've got the variants and we switch on the deleterious variants, then the coloring shifts to just show where those variations that have been identified as deleterious due to their kind of positional information, kind of where they actually live. So you can start to see different areas of the protein, and it gives you a little bit of information about them and the score as kind of predicted how sort of whether they're in one of these high confidence regions or not. So that's quite popular with our users. To kind of switch over a little bit and think about what's happening with LLMs. So lots of our services are exploring ways that LLMs can be used in different ways, typically around the literature summarization and assisted data curation. So one thing that we're doing in Ensemble within the vertebrate space, so we haven't taken it any further than that so far, is looking at gene symbol assignment. So it's something that people want to have descriptions of the genes, of course. And so before we were doing this on a kind of comparative genomics basis and transferring the symbols across, which is really slow. It's a quite intensive, resource-intensive way to do it. So now the Fergal Martin's team has developed this kind of approach whereby they trained for 30,000 symbols across 3 million vertebrate sequences. They had this transformer-based approach, and they were able to replicate the existing gene name assignments very well, and it takes about 40 seconds per genome, which is like a massive improvement on what we had before. And so then these are shown when you're in a gene, that we've got the description, and you can see this label here that it's come from this neural network prediction with a confidence. But we haven't pushed it. I mean, it works very well in vertebrates because vertebrates are very similar to each other, and so we haven't pushed it further yet, but that's something that we're interested in exploring. And then to go outside of Ensemble and to some of our resources that we link out to, so RNA Central has this nice pipeline called LitSum, which you can kind of access the reference down here. And what it does is it generates automated summaries for non-coding RNAs, which typically don't have that much information about them, using NLP techniques. So there's a piece of software called LitScan that goes through and extracts various kind of useful bits of information from different, like kind of the corpus, the literature. And then they feed all of those snippets to GPT, which then produces the summaries. So it's only working on kind of the things you've been given, and it's just to make it coherent and readable. There's some automated checking and refinement that kind of goes through a few loops to check that the summary is sensible. And every statement that's added is connected to the literature. So this is coming from the open access data in Europe PMC. So you can see here is a caution that it has been generated by AI with links that take you to understand how it's been generated. And then there's these PMC links for each of the sentences so that you can really go back and understand where it's come from. So at this point, it's the GPT is just being used to kind of organize the data rather than go away and find it for itself. But another couple of examples before I finish of different things that they're doing around this kind of functional space. So PFAM now has a new one called PFAM-N. So they've taken a neural network ProtCNN and trained it with various like 17,000 PFAM families. And this kind of this trained neural network then can make predictions as to the kind of the functionality of the different the different domains, and so it has been. They found after testing various things of the different ensembles of this neural network and then combining it with where they've been using Hammer as the approach before this hidden Markov model method, by combining the kind of where Hammer had high confidence and then where it had lower confidence using this ensemble of these protein neural networks, they actually got the best kind of accuracy of anything, of all the kind of different combinations they tried, and it's extended the coverage of PFAM by 9%, and it has actually been able to functionally characterize some of these domains which didn't have any predicted function before. And again, this was published in 2022. And then the last one to mention is the PROT-NLM, a natural language model that comes from Google. And so this will predict protein names from amino acid sequence. So it has this sequence-to-sequence model shown here represented here with the T5 in the middle of the schematic. So sequence data goes in and it comes out with a prediction as to what the name should be of that sequence. So the initial iterations were just kind of based on this, but then subsequently they've now started adding additional useful information, such as the species that the sequence is derived from, and also if it has an AlphaFold model, then that can be fed in too. That's helped improve the accuracy, and it's been applied to all the uncharacterized proteins in UniProt, and so that now has kind of increased the number of proteins which have a prediction using this approach. So there's lots of things going on in lots of different areas, but some of the challenges are the same, and which Raphael touched on some of these. So licensing can be an issue as to which software that you're using. So it's something that we have to keep in mind, and we need to make sure that everything that's generated is clearly labeled as such, particularly if it's going to be pulled automatically. We don't want AI being trained on AI generated content that might take us into a wrong place. So we want to make sure that that's all clearly labeled. Metadata standards for the AI methods are quite important because you kind of sometimes get things back from AI methods, but the metadata is not really captured in a very structured way. So that's something that will need to be kind of continually developed. Testing and benchmarking can be time consuming, and we need the right infrastructure to train and run the AI models. And also you need the AI expertise in-house. And as we're kind of using this across more areas, then it sort of requires more expertise. So that's something that we're all mindful of. The cost of running the models, commercial models, may become significant, and it also has the potential to drive up our carbon emissions. So it's something that we should consider. All right, so that was all I wanted to say. So just to acknowledge all of these people who are working on the different resources which I presented and which from whom I've pinched a lot of slides. So thank you very much.
Thank you very much, Sarah. Very impressive work at EBI. Is there questions? Q&A?
Right. Thank you, Sarah. Very interesting. The gene labels that are automatically generated with the neural nets, is that something that is going to be available to others for download, or?
Are these the ones in Ensemble?
Yeah.
So certainly, I mean, they're there and visualized on the website. So I imagine they're there in the downloads. I'd have to check. That's the vertebrates. As I say, it's only available for vertebrates at this time, but it's something that we need to test to see how far we can sensibly push it as we start to get to more diversity of organisms, like in the metazoa realm and such. So yeah, more testing needs to be done. But I think the data that's been produced, I imagine, is already available. But I can double check and let you know if you're interested in the vertebrate data.
Oh, well, I guess I would just wait for things to be rolled out to plants.
Okay.
But the percentage you showed in brackets of, is it like the accuracy, or it showed the label, then in brackets some information?
Sorry, no, I was talking before about the ensemble one. Sorry, the vertebrates one. So which one were you?
So if you go back to the first,
about the gene symbol
the
slide.
gene symbol.
Yeah,
this one. Yeah.
Okay.
Yeah.
So in brackets, the score there, is this like, what do you think? Is this accuracy or quality?
Confidence. I think this is a
Confidence.
confidence score.
Yeah, I think if you make that data available, would be nice to have it with that score, confidence score.
Mhm.
So the prediction is a description of this gene EPLP1.
Mhm.
Yes. Okay. It's very
Yes, so
very
this is
impressive.
taken from. So yeah, this is where this is one of the genomes has been generated from the kind of biodiversity project, so a vertebrate, and then typically they wouldn't come with a lot of information themselves. So this is where much of the kind of functional description will be assigned based on other better annotated vertebrates in the space.
So your quantity of data is enough to have something with a very good accuracy. It's 3 million sequences and train on 13
exactly 30,000
symbols.
sequences.
Okay. Oh, okay.
So I was asking kind of how many species that sort of we could push that down to that had high quality data, but it was in the realm of like 150 species or something was sort of what we're talking about. So yeah, there's quite a lot of data for vertebrates. Plants look on.
Right. Okay. So it could be a lot more challenging, you think, for plants. And how would you
with a mix of-
benchmark it?
So this is benchmarked against the kind of the orthology kind of previous tree building approach. So compared with how this one performed, and then evaluating the differences.
Yeah, I guess for plants, probably for like 30-40% of or more, we don't have a label or description. So if
Exactly.
the neural net is able to predict something for those, that would be nice. But how can we benchmark it? Does it need manual curation, you reckon, or?
I think typically with a lot of these methods, we kind of look to them for plants and then we say, oh, if only we had more curated data that we could use both for the training and the evaluation, right? So yes, I would say, I mean, if we have it, that's great. You need some kind of ground truth, right?
Okay, thank you.
Another questions to Sarah? Feel free to ask questions. There are a lot of very nice things presented, so if you have questions, feel free.
Sarah, I have another one? Say, how do you have a dedicated machine learning team, AI team at EBI, or do you have specific experts within each team? How do you structure expertise?
So we have a lot of research groups, particularly over in Heidelberg, and they are kind of doing a lot of the work around the imaging and developing kind of AI around that space and different ways that it can be incorporated. So it is more kind of whatever the research group is and their interest, they will kind of push in that direction. So it's kind of going in different directions. When it comes to the services, we don't have something centralized. We're sort of trying to level everybody up as much as we can to do things, which it's sort of the way it is at the moment. As we're looking to recruit, obviously it will be part of a thing that we're looking for when we're bringing new people in, because there's so much opportunity. And people come in with that experience. But also we're very happy to collaborate. A lot of this came from, collaboration with Google DeepMind or the AlphaFold work. So where there are opportunities to collaborate, and we can bring some of the data and the curation side, that's also quite a I suppose natural partnerships can form there. And we're very happy to collaborate. So yeah, if you have ideas and things that you would like to try out and we can help with, then happy to discuss. And I might not be the right person, but I can connect you with the with the appropriate team.
And the collaboration with Google DeepMind, it is a formal collaboration in the frame of the whole institute, or how do you work with them?
Well, so this was the specific example there was for the AlphaFold. So then it was a collaboration, a true collaboration, and then the data was all kind of launched on the day that the announcement was made. That's when the database went live. So obviously there'd been a lot of work together, kind of behind the scenes, to make sure that it was all positioned, ready to share. And so there's an ongoing relationship, I would say.
Okay. Is there another question to Sarah? If not, Kiwan, you can share your screen if it's okay for you.
Okay. Push it over there. Right, can you see it, Michael?
Yes, but it's not full screen. If it's possible to make it full screen.
I'm sure it's possible somehow. Then press slideshow.
Yes.
I've got just a few slides, and I wanted to be brave enough and do a demo. So
oh yes, good, nice.
So yeah, I think the slides are just setting the scene. Let me just press this button. Right, so I wanted to focus on a very specific feature we've been working on and trying to get it into production over the last probably 6 months or so. It's called the literature review feature of Netminer. So the idea is that when you're in Netminer, Netminer, you remember, is a gene discovery platform. You can search it with keywords, gene IDs. So you search for something like hypoxia, or you search for genes. What Netminer does so beautifully is it generates these knowledge graphs telling you a story about how gene, for example, is linked to hypoxia, right? Those knowledge graphs very often contain many publications. So the orange circles you see on the screen, they represent publications. And in the past, our end users spent a lot of time clicking on those edges, clicking on the publications, trying to get a feeling for, what is the story, extracting evidence, manually even copying things over into spreadsheets and so on from this view. Right, so we thought, okay, now with the emergence of language models, could we try to go from a graph that we generated based on a user query to a summary of that graph that looks a bit like what you see on the right-hand side. It's not an easy task to convert the whole graph into a textual representation. So we thought, let's start easy and just focus on summarizing the publications that are present in the graph. And if we tackle this and this is successful, then we can look into actually not just summarizing publications, but also summarizing relationships and other entities that are in a graph as a report. So we started this task and set out some key objectives that mattered to us. So number one was that we want to build a system that integrates knowledge graphs with language models. This is framed under RAG these days, so retrieval augmented generation. We use classical information retrieval to retrieve information from a database or knowledge graph, and then we feed that information to a language model to generate a summary. We want to use state-of-the-art technologies, and for now we said let's just use OpenAI and GPT models for our proof of concept. We were hoping with this approach to limit hallucinations because we are trying to ground the model in facts that are retrieved from the knowledge graph. It was like Sarah said in one of her slides, We wanted to provide citations, inline citations and references in the report, and also just generate a well-formatted report. Okay, so the workflow that basically I will demo in a second is looks a bit like this. This is our RAG approach, so retrieval augmented generation. The user comes to Netminer with a specific question in mind. They phrase the question, for example, list of genes, keywords. They get presented with a graph, which they can interact with. They can hide certain information from that graph. They can add more to it. And at some point they feel like, oh, this is interesting enough. Now I want to feed that to a language model and ask language model to summarize it. So there are steps around how do we rank which publications to send to a language model, because if a knowledge graph has hundreds, it would be quite costly to ask to summarize all. So how do we shortlist and find the most relevant publications for the summary? Then actually creating the summary, presenting it back to the user with the references and the links back to the knowledge graph. So the technology under the scene, it looks a bit like this. The user asks the question, the Netminer app uses a classical graph retrieval to query a Neo4j graph database, which also stores vector embeddings for all the publications. It will identify relevant genes and links to literature, feed that back to the Netminer app. That one prompts then GPT with the question and the literature, asks a GPT to summarize it. GPT provides a literature review, which we then combine with the rest of the knowledge graph and provide back to the user. And all this hopefully can be done within a few seconds, so the user doesn't need to wait too long. All right, so let's be brave enough and dive into a short proof of concept demo. I'll show you where we are currently with this idea. While I was preparing just the slides, I realized that probably never actually mentioned that we've been working on a new version of Netminer for the last year, and this is not released yet. It's stored under this URL called app-dev. So just a new user interface, looks all a bit fresher. So let's start and look into, so there are different ways of phrasing your search in Netminer. We could do a keyword search, a gene list search, or genome region search. So let's start with a gene list search and keep it simple. So we have a specific gene that we're interested in, TSN1. And for now, we have no idea what this gene is doing. So we are not going to combine it with any topics or keywords. We just search for TSN1 and want to know what information does the database, so our knowledge graph, hold about this specific gene. So it's telling us there are 4 copies of TSN1 in the knowledge graph, and it's linked to a number of different types of entities. So it's linked to 89 publications. It's linked to 4 different traits, plant ontology terms, and so on. So now let's select them and say, show us the evidence. So how is that gene linked to those pieces of information? So we press the Create Network button. So at this stage, we're using our classical graph retrieval to go and query the Neo4j graph and extract the links that connect those genes to the different types of evidence. And you can see that it's organized based on or which species the information belongs to. So what you see in this big box is wheat-related information. In this box, this is barley-related information. Some of the publications sit outside of a box because they're not clearly defined if they're barley or wheat, for example. So now, in the past, a user was presented a graph like this, and you can imagine how long it takes them to start navigating through this. So this is representing a gene. A gene is linked to a publication. This is the publication. It's linked to trait ontology terms, gene ontology terms. So first thing we did was, okay, we presented the user with a table view of the same information. That made it a bit easier. So this were the publications that are present in the graph. You see the year, you can navigate through it, but still it was not ideal. So now the next thing we wanted to explore is this literature review. So now let's see. If I go to the literature review button, use this, it's showing you an interface that is basically telling you in your graph, this graph is focused on the TSN1 gene, and we want to create a review with summarizing the information that are present in the 34 publications that are currently visible in the graph. Do you want the summary to be short, medium, or long? And here we go. So let's do that. So we press generate. We have a prompt in the background, and now it's going to get the abstract and titles from each of those publications, combine it with a prompt, send it to GPT, and wait for GPT to respond back. And then we present the review, the literature, what comes back from GPT in here. At the top, I think I need to zoom in a bit to make this a bit visible. Oops, too much. So at the top, it's saying, so with 34 publications, 15 of them were used. They are from this year range. And then the review starts with a general overview of this gene. Functional role, TSN1 is a sensitivity gene that interacts with TOX-A effector produced by several fungal pathogens. So we're slowly getting a feeling for maybe one or two functions that a gene is involved in. And inline citation. So you see that whenever it generated a sentence, it provides a link to the publication. If you click on it, it updates this info box, and it also highlights the publication in the graph. So this was a publication I clicked on. Now I can go and check actually how is this publication linked to my gene? Is it going through homology? Is it going through what type of path does it go and connects to that gene? So yeah, let's go back to the literature review, and I can say, yeah, okay, this is interesting. So I just want to see now if I, for example, ask for a more specific review that is focused around the topic of disease. But also I want to see if maybe this gene has any links to abiotic stresses. So let's say drought. And let's make it a longer review, generate again. So now we've got 70 publications that oh no, sorry, I only picked the 34. If the threshold goes over 50, it would start using the embeddings to find publications that are similar and then only pick a representative publication. So what I want to show you here now, because we provided a topic to the LLM. So this review is a bit more specific. So, and if we go down to this section here, which is relevance to disease and drought, while the primary focus of TSN1 is on disease susceptibility, its role in drought tolerance is less clear. However, the interaction with necrotrophic effectors may indirectly affect plant stress responses, including drought. And there's a paper cited here. And then over here, it also mentions there could be a link to drought. If we click on this, then we would now need to go and see if the publication is drought specific. I checked a few of them and they were actually. So this is the thing, you need to double check, see, is it hallucinating, is it not? But this is using now the GPT-4o model. And I feel like with every iteration of the models, they're getting a bit better. So this reviews and the network then at some point can be saved. If I close the literature review, I have generated 2 reviews, and I have, let's say, hidden a few pieces of information, added some to the, this is all interactive, and I don't want to go into all the details of how this people interact with this, but at some point they can say, I want to save that review and the knowledge graph all together as a bundle. So GSN1 report, there was drought and disease. Yeah, delete the bits that are not important, save. So this gets now saved into the user's workspace, and they can always come back to it, read the reports, or share this with a colleague. So for example, you see here, these are the projects I've been working on, and these are the knowledge graphs, how big they are, nodes and edges, and how many reports have been saved with this graph. And I can now take this and send it over to maybe my boss or my student and say, look, I've done some preliminary work here. Now take this over and add to it. So this was the first thing I just wanted to talk about it a bit and show you what how we've been trying to use language models in a very specific way to help a very specific task, which was slow in the past. Any questions about that before I move to the next one?
Yes, Sarah, do you want to ask the first question?
Okay, thank you. Kiwan, really nice. Well done for a successful live demo. I was wondering in the summary where you have, because obviously you have all the links out to the literature, which is completely excellent. Could you also have Go terms incorporated into the summary as well, so that they're, you have that kind of additional level of confidence in really what's being described, because you have them in your networks, right?
Right. So show the Go terms and actually link out the Go terms to maybe the node in the graph or to external websites. Is that sort of?
I was thinking a bit more direct, actually, whether they could be incorporated into the summary as you have the references in there, whether it was possible to connect in relevant Go terms such that if people pull the summary and parse it, they would have access to the Go terms. But I don't know, because that's maybe a level of indirection, which is perhaps a bit trickier.
Yeah, so, okay, this might actually bring me to my second topic, which I think relates to your question. So what we found is LLMs, if we ask the LLM to tell us which traits are mentioned in those publications, they would do a really good job. The moment we ask the LLM to tell us what the trait ontology IDs are of those traits, and that's when they start hallucinating and making up random trait ontology IDs, and probably same for gene ontology IDs. So the next thing I want to talk about is about how we are trying to tackle that challenge. So we feed the LLM a number of publications and ask it to extract the traits and trait ontology terms and IDs mentioned in those papers, which is interesting. Where I can switch to that if you want to.
Yeah, just I have a quick question before that. Yeah, do you plan to make a short note precising that it's generated by AI for your summary, or?
Yeah, I think we should mention it here somewhere in the summary, like Sarah had in her examples.
Yeah.
I think that little note at the top, I really like that. So yeah, it's nothing of this polished and finished yet. So it needs those notes to make it clear, and maybe which model we use, just a bit more information. I don't think we can provide any confidence.
No, I don't think so. Yeah,
that's really hard. Say how. So just making clear that it is AI generated, but so is everything really. I mean, this whole network is also AI. You can call it AI generated, right? We're not just doing a simple Cypher query. We are shortlisting the information we want to present to the user, right? If we go back to the actual to Netminer. Just give me a second. So where did we start from? Serious read. So we started with here. I searched for TSN1. I did a search. So at this stage, it's telling you this is the information that is linked to TSN1, 89 publications. But then once you say create a network, there are algorithms running in the background that shortlist what our algorithm thinks is the most relevant information and only presents that in the interactive network. Because sometimes we have cases where a gene is a hub gene interacting with hundreds of other genes. Each of those has 20 to 50 publications linked to them. So this number is going to the thousands of publications that can be somehow linked to a gene, directly or indirectly. So we have some algorithms that need to shortlist and rank. So I guess, that message of be careful, everything we're showing you here is based on some, AI or algorithms or computation is important across the whole platform.
But it's very impressive, yeah. And I also have a short question. Do you display a monthly limit? What is it? Well, yes, I saw that in your
for the review, right?
Yeah, a monthly limit. Perhaps you put a limit on something.
Yeah, so I mean, there's a actual cost associated to creating those reviews for us because we're using external APIs. Right? So each report has a cost. So so how we have structured this new Netminer, there is a free version, and under the free version, you get what the limit is we've set to. So if you're like a guest user, you don't even register, you come to this, you only get 1 or 2 reviews you can create. If you register, still for free, you get 5 or 10. And then if you subscribe to a premium account, then we give you 200 reports you can generate.
That's the 200 limit, okay.
So that's a limit you have there. Or it could be that you just say, I actually have my own API key. And then you provide your own OpenAI API key, and then you can it gets charged against your own credit card. Then you can use more, even as a guest user.
Yeah, very, very nice. Thank you.
Yeah, a lot of it is about thinking how these new things can be combined into a user-friendly interface as well. So it's user-friendly, not too overwhelming for the user. Which of the key features do we want to expose to users? Like, for example, temperature is one of the key parameters of GPTs. How to set the temperature. Is that something we want to expose to the users? Are there some other parameters? So yeah, we are still exploring and trying to, just like you guys, find out what works best.
I guess over time, as more and more people are familiar with using GPT, things like temperature will become more intuitive. Though you could kind of have like, an advanced user box and then they might like it. Whereas for now, that's probably a concept which most people are not familiar with, but it may become more established, right?
Yeah. Yeah, hopefully. Right. Just are we okay for time, Michael?
Yes. Let's go. It's very interesting. So
I can just talk about this topic a bit. So this is this paper illustrates the topic that I want to talk about and discuss with you guys. So this is based on the human phenotype ontology, but same applies for plant trait ontology. So literature curation often is about reading a piece of text and mapping terms to well-defined ontology terms, identifiers. All right. So like in this example, if you gave this sentence to a general LLM, it would most likely hallucinate and give you just a random human phenotype ID. If you use the classical, Lucene-based techniques to map this to a term or to a ontology ID, it would fail short when there are typos or things are slightly spelled in a different way than the synonyms you have in your ontology. So it has shortcomings there. If you have a text like this one at the bottom, expanded blood vessels, and you give it to a NLP algorithm, it would probably also fall short because it just can't really figure out that expanded blood vessels is the same as a vascular dilatation. Sorry, I'm not a biomedical expert. So the idea is if we fine-tuned LLMs, they would be able to overcome the shortcomings and also map things like this description to vascular dilatation and assign it this ID, right? So they've in this paper, they are basically saying it's possible, and they have used the OpenLlama, so open source language model, Llama, 8 billion models, parameters, and they have fine-tuned it with their ontology and saying they've got really great accuracies. So we tried to reproduce this a bit, and we weren't that successful. And maybe because we haven't had enough time, maybe because we don't have the proper GPU resources yet, Rossums did, or maybe we're just lacking the know-how how to fine-tune these open source models properly to make it all work. So maybe a combination of all of that. So it's been, we've been working on this probably for months or so, trying to make it work with my machine learning engineer. And yeah, we didn't really make much progress until just a few days ago, where we said, okay, just stop now this whole idea of using open source models, and let's just go back to ChatGPT and look into some of their new features. And one of them is called assistants. Have you come across assistants in GPT? So here we go. This is OpenAI Assistant, and in OpenAI Assistant, I can create a system, a well-defined system prompt like this one. Imagine your role is you're a biocurator, you're an expert in curation, and your job is to identify traits, and these are the steps you need to follow. Okay, so this we had previously as well, but still it was hallucinating with giving us fake IDs, fake trait ontology IDs. But then now with assistance, you can actually provide your own knowledge base as an additional source. So it's a bit like fine-tuning a model. So you provide your own knowledge base, and it also goes and takes that file and stores it and creates a vector base from that input. So we took all the trait ontology terms that are present in TO, 1,600 of them, and each of them became its own document stored in vector store, and said, okay, that's cool now. So let's try it. So that system prompt together with a vector search, and now let's go and grab a piece of text. So let's just take this one here and go back to the assistant, paste it in here. You can already see from the previous one that what you can expect to see. So I don't even need to prompt it anymore what to do, because it's all stored in that system instruction. Say running retrieval. Right, and then it generates this nicely structured JSON. So it's telling me the gene in this is MC1, it's barley, it talks about drought tolerance, and now this ID 276 Let's just double check this quickly. Trait ontology. Yeah, you can see this is actually drought tolerance now, yeah. So yeah, I've been looking at it for a few days now. I haven't had a lot of time to evaluate, but so far my feeling was like probably about 90% of the cases I looked at looked correct. Couple of times it didn't really get the completely wrong ID, but it maybe used a parent term, something that could have been a bit more specific or so in the ontology. So very impressed with it. So far so good. Problem, cost. To run this, we just tested it on 100 publications, wheat publications. If we use the GPT-4o mini model, the light, small, faster version, it cost about £1 for 100 publications, but that one was not as good. Missed some key information, got a few things wrong. So really the one I would love to use is this one, GPT-4o. On 100 publications, we estimated, and we have the dashboard, so we can see the cost was about £15 for 100 publications. So it's based on tokens that go in and out of that report, but about £15 for 100 publications. Now, in our knowledge graph, the one I was just showing you, we have about 200,000 cereals-related publications for which we would love to run this to extract this sort of information, gene trait information. So I haven't done the numbers, but yeah, it's not that cheap, and it's something that I possibly Marco was saying in an earlier meeting, how about we share the cost? Because maybe others would be interested as well in generating that sort of information. If we agreed on what is the key pieces of information we would want to mine for and extract from the literature, wheat or cereals related information, is there a way we could together apply for funding or create together, share as a resource maybe as well? We integrate into Netminer, others maybe into their knowledge graphs or databases. So I put this out for discussion and hear your views.
Thank you, Kiwan. It's very, very promising. Yes, I agree with you. If we pay for something, we have to pay in one place for the whole community, if possible. So it could be Netminer, or it could be EBI. I don't know where is the best place, but yeah, I think we will not have to duplicate this kind of costs. Mario?
No, thank you. I think all the presentations were very impressive, and I have a question. I will need to go in a minute because I have a visitor coming, but I wanted to ask a question before I go, if you don't mind. And it's looking into the future, and it's very relevant to my current kind of role in where we look at field trial data and crop performance data, because I wonder whether the next frontier could be bringing some of this information to combine to what it is clearly very strong underlying genotype to trait information. I have those traits perform in the field. Organizations like myself, well, we have data we have collected at INRAE, we have collected over many years. And I wonder if that could be a next frontier in terms of what we could do in this space. And I think clearly it would be very powerful. It's a question, a speculative question, but I wanted to ask it before I leave. Maybe Q1, if you, because you work as well in, you're very close to an organization in where you have as well a lot of crop performance data, which I mean field trial data.
Yeah, definitely. I think there's a lot of interest in that space at Rossumsted as well, Mario. Yeah, it's I think learning how to combine this the foundational models with your own custom knowledge and databases to build this fine-tuned language models. I think that's, and who to work together on it, or is everyone doing their own thing, are very important strategic decisions, because yeah, we all have the same problems in trying to hire the right people who have the knowledge, retaining them so they don't leave once they're picked up and are offered 5 times the salary from Google. Yeah, so yeah, I'm probably it's a good question, but I probably don't know what's the best way forward right now.
No, I think working together makes sense, and part of the WheatIS initiative mission is to make sure that we coordinate this work. I think that's something which we can actually take it up into the WheatIS information, the WheatIS initiative as something that we could propose. I think interesting with all this work you show us today, all of you, we started to have a role within the WheatIS initiative that is much stronger. It was always expected to come, if you like. We always talk about how important data is and how important the data for the community should be. But I think that's the first time we can now show tools that they will make that promise of an acceleration a reality. I think we clearly need to do more. That's why I'm thinking about that the next frontier could be bringing crop performance data. For me, it could be critical in terms of short kind of, I mean, there's still a lot of money that is gone from the connection between genotype and trait into how those traits perform in the field, right? And in particular for wheat, that matters more than maybe for a model organism because we are working clearly with the crop. But anyway, I mean, this is something that clearly we can take today a much more a stronger role within the WheatIS initiative in guiding the future, in particular coordinating what it could be funding calls. This is the type of thing in where it's much better coordination than trying to compete. I mean, clearly it's very obvious. Anyway, no, thanks for this. Sorry, I will need to go, but it's very good to see you all. And I think it's something we can take today very strongly as a message to the WheatIS initiative. Yeah.
Good to see you, Mario. But Marco just got on screen. He might have some views about field
No,
data.
I was just thinking that to me, we also still need data integration. There have been efforts in the past years, but so to make an example, if you want nowadays, if you want to make a query from farm to fork, as they say, so starting from genes, genotypes, and coming up to traits coming from field trials or information about food. You still need a lot of data integration, tailored data integration, while that's because we still are not using things like standards very much as yet. And if we could do more in that direction as well, applications like these ones that use artificial reasons, cutting-edge artificial intelligence could benefit as well.
I was going to say standards and metadata are
Yeah.
key here, and something EBI are passionate about.
Yeah, I know I work on that.
I think we all agree. I feel very, very strongly that could, if you're putting, for example, this type of tool in front of a breeder and you show them as well crop performance data, where you can link pedigrees, which is data we already have, that would be very powerful. I mean, of course, it's already very powerful, but you can see that the biggest challenge you have as the breeders will have is taking something that is performs well in a small control environment set up into something that will perform at scale. It still is one of the challenges we have in the sector. And
Could we work together, Mario, to just understand how to bring the crop performance data into a knowledge graph like Netminer has? And we can
that makes sense, Q. And I think clearly, because the organization where I'm working today, I'm leading today, we do have this because we have collected this from 100 years ago. It doesn't mean that data is 100 years old will be relevant today, but I think it's interesting when we are changing farming systems and we are looking into growing crops in a different way, reducing the inputs. And there are many of these old data sets that might be particularly relevant, and it will bring, it's almost that as well, the pipeline that goes from gene to farm. If like maybe we need to think in the other direction as well. But anyway,
okay.
Thank you very much.
Good
to see you all. Thank you.
See ya.
All right, I'm dropping off too. Bye, everyone. Thank you.
See you, Sarah. See you soon.
Bye.
You are mute, Michael.
Yes, thank you, Raphael. Is there another question to Kiwan? It's okay for you? Okay, so I think with just these three presentations, we had a very interesting overview of what you are doing on AI, and I'm very impressed on what has been done at Netminer, also at EBI. So yeah, and I agreed with Mario. I think we will have a big role in the future of wheat initiative. I think the wheat researcher and wheat breeder will ask us a lot of things, and to have some tools using AI to tackle the current questions. Okay, is there any other comments? If not, I will stop the recording. Okay, so thank you. Thanks to all of you, and I hope to see you at PAG. And we'll have the two remaining presentations from Green Genes and from Australia. And so it will be a good occasion to continue to discuss about that.
Thank you.
Thank you very much.
Thank you.
Bye.
Thank you.
Bye.